Hello students, welcome to EPG Pathshala. I am Dr. Gulshan Kumar Bansal from Department of Pharmaceutical Sciences and Drug Research, Punjabi University Patiala. Today we are going to discuss about a module Medical Nanobiotechnology and Nanoanalytics Diagnostic Tools under paper Nanobiotechnology. The objectives of this module include the diagnostic techniques for early detection of diseases, particularly chronic and life-threatening, have been eluding the medical practitioners for a long time. Nanobiotechnology has offered numerous tools for early diagnosis of many complex diseases such as cancer, Alzheimer's disease, liver disease, cardiac disorders, etc. Now, this presentation provides a comprehensive information on composition, applications of various types of nanostructures as diagnostic tools. Medical science has witnessed exponential growth in facilities to accelerate the disease diagnosis, to provide targeted drug therapy, to improve pharmacokinetics of the drugs, to increase specificity of the drug treatment, to minimize the surgical interventions, to identify relevant biomarkers, and to improve tissue imaging techniques for a specific medical treatment of a disease or disorder. Most of these recent advances are attributed to the advent of nanotechnology. So, when nanotechnology is applied in biological system, it is termed as nanobiotechnology. The various nano-sized materials have brought revolutions in diagnosis as well as medical and surgical treatments. So here, let us first have a short preview of various applications of these nanomaterials in different fields of medical science. In the first kind of application in the surgery, gold nanoparticles have been exploited for removal of lipids from the body. Many health problems arise from obesity. In most obese patients, diet control and even anti-obesity drugs do not provide the required results. So in such cases, liposuction is carried out. It is actually a surgical procedure to remove the excess of fat from a selected part of the body. Shang et al. Have conducted an experiment on nanoparticle-assisted liposuction. They injected a solution of gold nanoparticles or gold nanorods into the fatty tissues to be removed, as shown in the first picture. The nanorods, being lipophilic in nature, diffuse into the fatty tissues uniformly, as shown in the second picture. In the third picture, it is shown that the infrared laser beam is targeted on the tissue treated with the nanorods. The nanorods, being made up of metal, heats up and cause melting of the fat. The molten fat is finally suctioned out with the help of a needle, as shown in the last picture. So the nanorods have assisted in removal of unwanted fatty tissues with any need of open surgery. Nanobiotechnology has been used in fabrication of dental implants. The major problems with the purely metallic implants are their hardness and immunosensitivity. These limitations are overcome by fabricating the implants from nanomaterials. These are not only extremely strong without being too hard like metals, but also do not trigger any immune response because of functional coatings on them. Another application of the nanomaterials in the disease diagnosis. Disease diagnosis is the most important part of medical treatment. When any pathological condition occurs in the body, the affected tissue undergoes some biochemical changes. The clinical diagnostic tests actually see those biochemical changes through blood or other biological samples. The most reliable and most efficient diagnostic method is to directly visualize the affected tissues. Nanoparticles have brought significant advancement in this area, particularly in the diagnosis of cancer, which is otherwise diagnosed at a very late stage. In the first step, a dispersion of magnetic nanoparticle is injected into the body, as shown in the first picture. Secondly, it is distributed evenly in the body. Then depending upon the surface protein specific to the cancer cell, these get localized in the cancerous cells as shown in the third stage. Now here they play multiple roles depending upon their fabrication. If the location of tumor is to be ascertained, the body is scanned through magnetic resonance imaging. The concentrated nanoparticles, being magnetic in nature, will be easily detected and location of the tumor is identified. The other roles are that they release a specific drug in a controlled manner, or they cause hyperthermia upon incident of low intensity infrared radiation to kill the cancer cells without affecting the normal cells. Molecular imaging is another field in medical science. It is defined as in vivo visualization, characterization and measurement of biological processes at the molecular and cellular levels. So, nanomaterials, they assist in easy diagnosis of disease states or disease tissues without any surgical intervention. Molecular imaging is an important technique or important step in the diagnosis of a disease. The different types of nanomaterials, we call them nanoprobes, they are actually miniature machines which can attach themselves to particles in the body, for example antibodies, and they emit a magnetic field. Now probes that are not attached to anything, they do not create any detectable magnetic field. So this magnetic field is the key factor in molecular imaging by the nanoprobes. So nano tracking may be able to detect tumors that are even a very few cells in size. The different types of molecular imaging modalities which are used for molecular imaging are positron emission tomography, we call it PET, single photon emission computer tomography, we call it SPECT, magnetic resonance imaging, we call it MRI, as you all are knowing it, then contrast enhanced ultrasound, that is a CEU, then we have optical bioluminescence and optical fluorescence. Now these are the different techniques which can be used standalone, or they can be used in combination also. For example, PET CT scan is used in a diagnosis of or images of the cancerous tissues. Similarly, we have the SPECT CT scans which are used for the same purpose, and we have the PET MRI which is even more sophisticated than the PET CT scan for the cancer images. Tissue engineering is another important field in medical science. Nanomaterials are fabricated as scaffolds for housing, for providing the conducive in vivo like environment for growth of cells to develop tissues or organs. These cells may be stem cells or cells isolated from a living system for a specific organ. The tissue or organ grown in vitro under the simulated in vivo conditions provided by the scaffolds are used in repairing the damaged tissues or organs. This concept is not limited to cardiac repair as shown in this picture. Artificial skin is also grown by similar kind of technique which is used in burn cases. Nanomaterials are of different shapes and compositions, and these are fabricated to meet specific purposes. The different types of nanomaterials on the basis of shapes are nanoparticles, nanowires, graphenes, and cantilevers. The nanoparticles are spherical bodies fabricated from different inorganic and organic chemicals. Accordingly, these are classified as inorganic nanoparticles and organic nanoparticles. According to the material used for its construction, they are called gold, silver, iron, copper, silica, and in general metallic nanoparticles under the inorganic category, and organic nanoparticles include dendrimers, micelles, liposomes, and ferritins. Quantum dots are also a type of specialized nanoparticles. Cantilever is a flat strip of silicon carbide which is flexible. Nanowires are very thin fibers composed of carbon atoms. Graphene is a single atom thick sheet of carbon atoms. It is very light but very rigid and much stronger than steel. It has a very good thermoconductivity. Rolling of a graphene sheet at a certain angle gives a tubular shaped nanomaterial called nanotube. From these different types of nanomaterials, Nanoparticles, quantum dots, and cantilevers are being used extensively in disease diagnosis. So here, the general method of synthesis of and working of nanoparticle is displayed. Firstly, the nanoparticles are prepared. Here we have taken the example of an iron nanoparticle. Then it is functionalized with polyethylene glycols to make them hydrophilic as well as to make them versatile for appendages of suitable bioactive molecules. In step 2, these dispersed nanoparticles are treated with the suitable bioactive molecules like proteins, oligonucleotides, antibodies, and any other biomolecule to make the nanoparticles biofunctionalized. In step 3, the biofunctionalized nanoparticles are injected into the cells. Inside the cell, they bind with a specific target and get localized. Finally, the cells are scanned through appropriate imaging techniques like MRI or other scanning techniques to visualize the cells through detection of the localized nanoparticles. So, a variety of polyethylene glycols are available to coat the nanoparticle surface. A specific polyethylene glycol is chosen to append a specific biomolecule on the nanoparticle, and this selection is dependent upon the kind of functional groups present on the biomolecules which can bind with the polyethylene glycol. So here is a general diagram of a nanoparticle. It may be impregnated with drugs as shown here, or radioisotope materials or genes. These drugs or biomolecules or any other specific ligand can be coated on the outer surface of nanoparticle. A nanoparticle may be designed to carry more than one drug or targeting ligand. Here is the case of iron nanoparticle and superoxide nanoparticles. Functionalization of these nanoparticles with a specific radionucleotide, for example copper 64, indium 111 and iodine 124, is used in imaging of different tumor cells, they are used for the tumor angiogenesis, they are used for tumor visualizations or imaging of mesothelioma or imaging of breast cancer using optical techniques. Gold is one of the most stable metal. At the same time, it is malleable and ductile. Due to the appealing properties of the size controllability, good biocompatibility and easy surface modifications because of the inherent properties of the gold, it is an attractive alternate for the construction of imaging agents. Gold nanoparticles exhibit unique optical properties such as distinctive extinction bands in the visible region because of the surface plasmon oscillation. The fluorescent color exhibited by these gold nanoparticles is dependent on their size. Small size gold nanoparticles, which are about 30 nanometer, exhibit blue fluorescence, and as the size increases, the fluorescence wavelength increases and it appears red as shown in the figure. On the extreme left, there is a red colored gold nanoparticles, and on the extreme right, they are the blue colored gold nanoparticles. So we have seen the gold nanoparticles in the in the detection of the cancer. So now let's see a short video of gold nanoparticles, how they are prepared and how they are used in the detection and treatment of cancer. So here is the video.
What if we could pinpoint the spread of cancer earlier and take some of the uncertainty out of diagnosis and treatment? That's the goal of Massachusetts General Hospital researchers Ralph Weissleiter and Mukesh Harrisangani. They are targeting the body's lymph nodes. Many cancers spread to lymph nodes first, but we don't currently have very good ways of seeing if small tumors have begun to grow in them. Dr. Weissleiter designed tiny nanoparticles made of iron oxide and coated with dextran, a sugar-like substance. This coating causes the nanoparticles to be taken up by normal cells in the lymph nodes, but not by any cancer cells that may have taken root there. A magnetic resonance imaging machine reveals the difference. The researchers hope their new technique will improve cancer diagnosis and lead to earlier and more effective treatments.
When tumors are first diagnosed, the first key question for the treating oncologist is whether this primary tumor has already shed its cells to distant locations within the body.
The only criterion we've had for determining whether a lymph node is involved is by the size of the lymph node.
In other words, if anything is small, we tend to ignore it and call it something that is normal. Whereas if something is bigger or enlarged, then we tend to call it tumor.
That doesn't sound very precise, and it's not very precise, but it's the best we've been able to do.
In order to get a more precise diagnosis, doctors would need to find a way to see inside the lymph nodes. So Dr. Weisslieder set out to design special iron oxide nanoparticles that could be used to enhance MRI images.
So the particles in these bottles are the nanoparticles. They are small in size. They are essentially rust particles. That's why they are called, iron oxide or nanoparticles. These nanoparticles come as a powder form. And what we do essentially is mix them with fluid. And this is how they look like once they are in solution. And then we take the amount that is required to be injected based on the patient's weight and then inject these particles. Once the patient comes in, they would get an MRI without the nanoparticles. And then after the initial MR, they would be injected with the nanoparticles and would have to return 24 hours later. At that point, they would get scanned again. And at this point, if the lymph nodes are normal, then they would take up the nanoparticles and turn dark or become black. Whereas if they are filled with cancer, they would not take up the nanoparticles and stay bright.
In one of the largest prostate cancer trials, we imaged over 80 patients and systematically analyzed over 350 individual lymph nodes. And what we observed was stunning. We had a sensitivity or accuracy for detection of lymph node metastasis well over 90%, which was really unprecedented.
So it was a very exciting thing to sort of be able to pick out these small little areas of tumor infiltration, which beforehand was not practically possible with what we have available.
If the nanoparticle technique is widely approved, it'll make a profound difference in the way physicians make clinical decisions, and will make those decisions more exact and, I believe, of tremendous benefit to patients.
Okay, Dr. Kaufman, so this patient with prostate cancer that you refer to us for nanoparticle exam, on the pre-contrast study, you can see that there is an enlarged node in the pelvis. And if you look at the size, it's about 1 centimeter by 2.3 centimeters. So by size, it is malignant, and it should be considered suspicious for tumor. But interestingly, after we gave the nanoparticles, you can see that the node has turned homogeneously dark, which is a sign that the node is actually normal and benign, rather than it being malignant.
We can now say that this is a normal lymph node, despite the fact that it's enlarged, and that changes the management completely in a much more favorable way for the patient.
Now, gold nanoparticles are available in different sizes, from the very small to the quite large one. When we say the large one, it means not more than 100 nanometers. So the gold nanoparticles, which are having a size of 100 to 10 nanometers, they primarily accumulate in all the organs of the reticuloendothelial system. And where the gold nanoparticles, which are having a size in the range of 15 to 20 nanometers, they may cross the blood-brain barrier or even the blood-retinal barriers also. And the gold nanoparticles, which are having a size less than 2 nanometers, they are highly toxic because they can penetrate any part of the body, any part of the cells, and there is a possibility of irreversible binding of some key biopolymers. Quantum dots are other type of nanoparticles which are made up of a semiconductor material, usually selenides or sulfides of metals like cadmium or zinc within a range of 2 to 10 nanometers. So it consists of a metalloid crystalline core and a cap or shell that shields the core and render the quantum dot biocompatible. Biocompatible coatings or functional groups are added to the core shell to improve quantum dot core durability and suspension characteristics and to achieve a desired target such as drug delivery or molecular imaging. Here it is important to note that any compromise on the coating material or process may expose the metalloid core in vivo, which may be toxic either as a composite core or upon dissolution of the quantum dot core to constituent metal, for example, cadmium. Now, the property of the quantum dot which makes it suitable for a diagnostic agent is the semiconductor crystals they fluoresce when stimulated by UV light of a specific wavelength. The wavelength of the fluorescent light emitted from the crystal depends upon the size of the crystals, as shown in the diagram picture below. The laser light, it excites the quantum dot and it releases some light which is marked as emission, and this emission is detected to image the tissues. Smaller quantum dots which are having a diameter of less than 2 nanometers gives a blue fluorescence, while larger particles which are having a diameter of up to 5 nanometers, they give red fluorescence. The quantum dots with a size of more than 5 nanometers can be rapidly cleared from the kidneys. Due to this broad wavelength range of excitation and narrow and symmetric emission spectrum properties, quantum dots are well suited to optical multiplexing, in which multiple colors and intensities are combined to encode genes, proteins, and small proteins libraries for imaging tumors.
Everybody, and welcome to the Quantum Dot Multiplex Theater. Today, Lil and I are going to tell you about our exciting future in nanotechnology. By the way, Lil, what would you like to be when you grow up?
Gee, Dot, I don't know. What are my choices?
Well, there are already a lot of quantum dots in the clothes people wear, sports gear, solar power cells, and a lot of us are even getting into healthcare these days.
Now that sounds interesting. I'd like to help people be healthy. How can I do that?
Well, let me see if I can make it easy for you. We're nano-sized particles that have very special, downright surprising characteristics at our small size.
How big is nano?
Well, we can't be seen with the naked eye, or even with the kind of microscope that you have in your school science lab. Let's see. I'm about 7.2 nanometers wide, and if you lined up about 3 million red quantum dots like me, side by side, they would be about the width of a human thumb, or about 1 inch.
Wow, that's really tiny. What are we made of?
Quantum dots can be made of various elements, but our tiniest building blocks, our atoms, are arranged in crystal formation. That means they're put together in organized, repeating, geometric three-dimensional patterns. And we're semiconductors.
Great! I'd love to drive a train.
Well, no. What that means is that we're excitable. And when we get excited, we emit energy, which can make us glow.
So that's why I glow sometimes. Well, why are you red and I'm blue?
Think of it this way. A semiconductor is in a little box, and if it's squeezed down even more inside that little space, its energy changes. As for quantum dots, depending on our size, we can give off light energy in a full range of fashion colors. Maybe watching this movie will help.
cool.
I certainly think so, and so do healthcare researchers.
So how can I help people feel better?
In a lot of ways. You see, we're very clever. For example, we can play James Bond here in the nano world. By attaching us to certain biological tracking devices, like antibodies, researchers essentially program us to find specific cells that are sick.
You mean like cancer cells?
Yes, that's right. And when they use different colored quantum dots at the same time and program them to attach to different things they want to observe, researchers can see how molecules interact inside cells. And then they know more about biological processes inside the body. This multicolored technique is called multiplexing.
That certainly is clever. But how do we get inside cells?
Depending on what they want us to do, researchers give us a special shell or coating that works like a molecular key to get us inside cells where we can do good things. We can deliver drug treatments or genes, and this is the really cool part. When we're built to work in sick cells, we can treat them without hurting any of the healthy cells. Just watch this. So here it is shown that how a quantum dot conjugated or functionalized with an antibody specific for prostate cancer binds with the prostate specific antigen and get attached to the membranes to facilitate diagnosis of the prostate cancer. Now the growing concern of toxicity associated with the heavy metals, specifically cadmium, nickel, lead, mercury, some of these are used in quantum dots, have guided various legislations across the globe to recommend for prohibition of use of these metals in fabrication of anything related to household appliances, sports, leisure or any other such purposes. On the basis of same backgrounds, quantum dots are being fabricated without cadmium, which are called cadmium-free quantum dots. The latest trends are now in the fabrication of silicon-based quantum dots. This picture is showing silicon quantum dots fluorescing inside the cancer cells.
It might someday be akin to gulping Pepto-Bismol with incredible results. Researchers say quantum dots, ultra-tiny chunks of superconducting crystal, might help doctors identify tumors or problem microbes in your body early on. The dot's small size would allow them to cling to protein or enter cells, thus becoming a useful medical imaging tool. There are problems with using the tiny crystals. Quantum dots can break open and release toxins, especially when exposed to UV light. But a new study published in Nature Nanotechnology says tests on primates have been successful. Before real use begins, scientists will need to further study how quantum dots travel through and interact with major body organs. Granted, swallowing quantum dots sounds a bit scary, but hey, it's better than injecting us with Dennis Quaid, right?
The next type of nanomaterial is dendrimers, which are a group of highly branched spherical polymers and that are often referred to as artificial proteins. A monomer is polymerized in a controlled fashion around its own core to generate layers of monomers in the form of a branched polymeric network. The number of layers define the generation of dendrimer. Like here as shown in the figure, a G4 means a 4th generation dendrimer has 3 layers of polymerized monomers around the core monomer. By controlling the degree of polymerization, dendrimers can be constructed in various sizes, molecular weights, and chemical compositions due to high loading capacity of payloads and the capability to control the polymer structure. Dendrimers are a favorable platform for the construction of multimodal imaging probes. The various properties of dendrimers include that they have a nano size scale, means their size is very small. They have the monodispersity, means when they are dispersed in ecosystem, individual molecules are separated. They are quite viscous, and they have a very high equisolubility, so there is no problem in the dispersibility in the biological system, and at the same time they have a very high solubility in the non-polar solution. So if you want to fractionate, we can do it, and they are non-crystalline and they have the low compressibility. So here it is shown that how the drug is loaded in dendrimers. The figures A to C show that the drug molecules first align themselves around the periphery of the dendrimer, and then they diffuse inside the dendrimer and remain there as individual molecules. Alternatively, as depicted in figures D to F, the biomolecules align weakly around the dendrimers. Then many such dendrimers come closer to encapsulate the biomolecules inside their network. The various forces responsible for these attractive interactions are electrostatic and covalent interactions. High density of amino acids or carboxylic amino function groups or carboxylic function groups on the surface of dendrimers enhances the solubility of hydrophobic drug by electrostatic interactions, whereas the large functional groups on the surface of dendrimers are responsible for covalent conjugation of numerous drugs with the relevant functional groups. Micelles are another type of organic nanomaterials used in diagnosis. These are self-assembling colloidal nanoparticles with a hydrophobic core and hydrophilic shell composed of surfactant like sodium lauryl sulfate or different types of toluenes. These are functionalized with polymers and targeting molecules and may be drugs too. These can passively accumulate in the areas with leaky vasculature such as tumors, inflammation, and infarction. In pharmaceutical development, micelles have been successfully used as carriers for water-soluble drugs. Recently, polymer micelles are gaining an increasing intention for tumor imaging due to their high stability and good biocompatibility. A special group of polymeric micelles can be synthesized by the conjugation of soluble copolymers with lipids. The modified micelles can carry various specific target moieties on the surface, which may be suitable for imaging probes for multimodal tumor diagnosis. Now, cantilevers is another type of nanomaterial, which is a very thin, flat, flexible strip of silicon carbide coated with the probe molecules for a specific target molecule. When a biological sample containing target as well as the other interfering non-target molecules are passed through the cantilever as shown in the figure A, the target molecules conjugate with the probe molecules as shown in figure B. As a result, the weight of the cantilever is increased and it bends. The extent of bending is quantitatively related to the concentration of the target molecules in the sample. So this picture is showing how a cantilever loaded with a specific antibody can detect the marker proteins from a specific cancer cells. Like the blue colored antibodies, they are detecting the blue colored antigens released from the cancer cells, and red colored antibodies, they are binding with the red colored proteins being released from the cancer cells. So how this cantilever helps in detection? Here it is explained how the binding is detected. A beam of light which does not interact with any component of the cantilever pro-molecule or target molecule is incident on the cantilever, it is reflected to reach a detector. When cantilever bends due to binding, the angle of reflection changes and hence the direction of reflected beam also changes. These optical changes are then converted into a quantifiable signal. So finally, in summary, we can say that the diagnostic techniques in medical science have grown to a new level with the advent of nanotechnology. Due to extremely small size, the nanomaterials can penetrate deep into all biological components. They can made to cross even blood-brain barrier. Diverse functionalization of the surface of the nanoparticle with a variety of biomarkers such as specific antibodies, oligonucleotides, peptides, fluorophores, they help in defining their specificity. So it is said that a disease diagnosed in time is cured. So nanomaterial based diagnostic tools help in early detection of the diagnosis of any pathological state. Thank you.